Hello everyone. So today we are going to discuss microarrays. A microarray is a multiplex lab on a chip. Its purpose is to simultaneously detect the expression of thousands of genes from a sample like a tissue. It is a 2D array on a solid surface, usually a glass slide or a silicon thin film cell, that accesses large amounts of biological material. The concept and methodology of microarrays was first introduced and illustrated in antibody microarrays, also referred to as antibody matrix in 1983. The gene chip industry started to grow simultaneously after 1995, with the establishment of companies such as Affymetrix, Agilent, Applied Microarrays, Arrayjet, Illumina, and others. The technology of DNA microarrays has become the most sophisticated and the most widely used, while the use of protein, peptide, and carbohydrate microarrays is rapidly expanding. A microarray is a laboratory tool used to detect the expression of thousands of genes at the same time. DNA microarrays are microscope slides that are printed with thousands of tiny spots in defined position, with each spot containing a known DNA sequence or gene. Often these slides are referred to as gene chips or DNA chips. The DNA molecules are attached with each slide act as probes to detect gene expression, which is also known as transcriptome or set of messenger RNA, transcripts expressed by a group of genes. Look at the bottom right hand side and see the picture of a microarray chip. Microarray is one of the most recent advances being used for cancer research. It provides assistance in pharmacological approach to treat various diseases, including oral lesions. Microarray help in analyzing large amount of samples which have either been recorded previously or new samples. It even helps to test the incidence of a particular marker in tumors. Till recently, microarrays usage in dentistry has been very limited, but in future, as the technology becomes affordable, there may be an increase in its usage. Once the human genome sequence was completed in 2001, it paved the way for many experiments and researches. One such area was identifying the region of DNA which control normal and disease states. Functional genomics is the study of gene function through parallel expression measurements of a genome. The most common tool used to carry out these measurements includes complementary DNA microarray, oligonucleotide microarray, or serial analysis of gene expression, that is SAGE. Prevention, diagnosis, and treatment in dental practice are based on an understanding of the biology of underlying oral health and disease. Few aspects of a patient care will remain untouched by today's rapid advances in biological research. In the future, dentists may use inexpensive but remarkably sophisticated diagnostic tests to diagnose infection, oral lesions, and symptoms of TMD. The small variations in the DNA sequence that lead to different characteristics such as skin color, facial features, or height are known as polymorphisms, which also can cause or contribute to the development of many syndromes and diseases. These genetic variations can be easily identified by the microarray technique. Microarray provides a basis to genotype thousands of different loci at a time, which is useful for association and linkage studies to isolate chromosomal regions related to a particular disease. This array also can be used to locate chromosomal aberrations related to cancers, such as segments of allelic imbalances, which can be identified by the loss of heterozygosity. By comparative genomic hybridization techniques on genomic DNA, amplified or deleted regions in the chromosome can be identified, such as in the case of oral cancer. To perform microarray analysis, mRNA molecules are typically collected from both an experimental sample and a reference sample. For example, the reference sample could be collected from a healthy individual and the experimental sample could be collected from an individual with a disease like cancer. The two mRNA samples are then converted into complementary DNA, and each sample is labeled with a fluorescent probe of a different color. For instance, the experimental cDNA may be labeled with red, whereas the reference cDNA may be labeled with green fluorescent dye. The two samples are then mixed together and allowed to bind to the microarray slide. The process in which the cDNA molecules bind to the DNA probes on the slide is called hybridization. Following hybridization, the microarray is scanned to measure the expression of each gene printed on the slide. If the expression of a particular gene is higher in the experimental sample than in the reference sample, then the corresponding spot on the microarray appears red. In contrast, if the expression in the experimental sample is lower than in the reference sample, then the spot appears green. Finally, if there is equal expression in the two samples, then the spot appears yellow. The data gathered through microarray can be used to create gene expression profiles, which show simultaneous changes in expression of many genes in response to a particular condition or treatment. Gene microarray technology rests on the ability to deposit many tens of thousands of different DNA sequences on a small surface, usually a glass slide, often referred to as a chip. The different DNA fragments are arranged in rows and columns such as the identity of each fragment is known through its location on the array. Two types of microarrays are gene expression microarray and tissue microarray TMA. Techniques like northern blot and reverse transcriptase polymerase chain reaction allow testing for only a few genes per experiment. But microarray or global expression profiling not only looks at orders of magnitude of more gene than that was previously possible, but also has the advantage that the genes examined are not influenced by the pre-selection of genes. Now let's see the principles of microarray. mRNA is an intermediary molecule which carries the genetic information from the cell nucleus to the cytoplasm for protein synthesis. Whenever some genes are expressed or are in their active state, many copies of mRNA corresponding to the particular genes are produced by a process called transcription. These mRNAs synthesize corresponding protein by translation. So indirectly, by accessing various mRNAs, we can access the genetic information or gene expression. This helps in the understanding of various processes behind every altered genetic expression. Thus, mRNA acts as a surrogate marker. Since mRNA is degraded easily, it is necessary to convert it into a more stable cDNA form. Labeling of cDNA is done by fluorochrome dyes, such as CY3 and CY5, green and red respectively. Picture on the bottom right-hand side. The principle behind microarray is that complementary sequence will bind them with each other. The unknown DNA molecule are cut into fragments by restriction endonucleases. Fluorescent markers are attached to these DNA fragments. These are then allowed to react with probes of the DNA chain. Then the target DNA fragments along with complementary sequences bind to the DNA probes. The remaining DNA fragments are washed away. The target DNA pieces can be identified by their fluorescence emission by passing a laser beam. A computer is used to record the pattern of fluorescence emission and DNA identification. This technique of employing DNA chip is very rapid, besides being sensitive and specific for identification of several DNA fragments simultaneously. The study of the expression of most, if not all, genes in a specimen is not hypothesis-driven, as most of the studies used to be, but is instead referred to as discovery-type research, or in the less flattering description as fishing expeditions. Whereas cDNA derived from a tumor hybridized into a chip to study gene expression levels. Alterations in DNA copy number can be measured by hybridizing fluorescently labeled DNA from a tumor specimen to these chips. TMAs are constructed by transferring cores of paraffin-embedded tissue to pre-cored holes in the recipient paraffin block. Over 500 cores can be placed in a single block by this technique. Sections cut from TMA block can be then used for immunohistochemistry or in situ hybridization studies. TMAs are similar to gene expression microarrays in having sample arrayed in rows and columns on a glass slide. They differ in each element of the TMA slide corresponding to a single patient sample, allowing multiple patient samples to be assessed for a single molecular marker in one experiment, while gene expression array allow assessment of thousands of molecular markers on a single patient sample per experiment.
I am SAMBIT MAJUMDAR and my topic is on principle and application of microarrays. I will mainly focus on types of microarrays and tissue microarrays. Types of microarray. First is DNA microarray, which is a tool used to determine whether the DNA from particular individual contains mutations in genes. There are four types of DNA microarray: cDNA microarray, oligo-DNA microarray, backed DNA microarrays, and SNP microarray. Next is protein microarray. In this, it is a platform for characterization of hundreds of thousands of proteins in a highly parallel way. Next is peptide microarray. These microarrays are used for detailed analysis or optimizations of protein-protein interaction. Next is tissue microarray. This type of microarray are used mainly in pathology. Next, cellular microarray, which is also called as transfection or living cell microarray, and are used for screening large-scale chemicals. Next, antibody microarray, which are used for detecting of antigen that are protein-specific microarray. Next is phenotype microarray, which is mainly used in drug development and they measure cellular phenotypes. Next is reverse phase protein microarray, which is used in clinical trials in the study of biomarkers and many others. And the last is carbohydrate microarrays, which are also called as glycoarrays, can be used in protein binding affinities. Tissue microarray. Tissue microarray is a method for organizing and analyzing tiny amounts of tissue samples on a solid support structure. This microarray technique was originally described in 1987. It is a high-throughput research tool which has greatly facilitated and accelerated tissue analysis by in situ technologies. They are used to every research methods that can be applied on standard whole sections at enhanced speed. Tissue microarray has molecular targets can be DNA, RNA, proteins, depending on the type of probes used for the analysis. This type of technology may be specifically applied to the profiling of proteins and biomarkers of interest in other pathophysiological conditions such as cognitive heart failure, hypertension, diabetes, cystic fibrosis, and many others. This technology has the capacity to allow quantitative assessment of change in protein markers across a range of tissue samples from multi-center toxicological studies. How tissue microarray works. Paraffin blocks produced by extracting cylindrical tissue cores from different paraffin donor blocks and re-embedding this into a single recipient block at defined array coordinates. Using these techniques, up to 1,000 or more tissue samples can be arrayed into a single paraffin block. Tissue microarray devices principally consist of three key components. First is the hollow needle, which is used to produce hole in an empty paraffin blocks. Second is again the hollow needles with a slightly thinner diameter from the previous one to punch a tissue core from the tissue block and to release the tissue core into the pre-made holes of the recipient block. A micro-manipulated XY stage that moves the recipient blocks to the predefined coordinates. Methods of tissue microarray. First is extracting the cylindrical core tissue from a region of interest in a donor. Then the samples are deposited into a recipient paraffin blocks. After that, coordinates XY of each core are tracked using a relational database. Microtome sectioning to generate 5 micrometer thick slides for molecular analysis to charge microscope glass slides. Then the microscope and image analysis is done. At last, the data is stored in a database and clinical database. result, which is taken from one of the articles in which they have used tissue microarray for their analysis. Diagram with capital A labeling showing eosin strained cores from normal human kidney cortex and the label capital B showing the medulla and the label capital C showing a prostate. Advantages of tissue microarray. First is amplification of scarce resources. Second is simultaneous analysis of very large numbers of specimens. Decrease in assay volume, time and cost. And the last is original block does not get destroyed for diagnosis and thus conserves valuable tissue. Application of tissue microarray. First is diagnostic biomarker. Diagnostic biomarker can serve as clinical studies as screening tools for certain diseases or for identification of specific subclass in a disease. Second is non-neoplastic tissue microarray. This is a study in other non-neoplastic tissues like dermatological, cardiac, and placenta diseases. Third is prognostic biomarker. These are generally based on the study of carcinoma of oral cavity. Fourth is clinical application. It is used in pathological department for testing of new antibodies and probes or determining optimal staining condition using a small test of tissue microarray. Main objective is to define the functional status of the tumor prior to the therapy. Last is the quality control. In quality control, tissue microarray can facilitate the immunohistochemistry staining procedures and interpretation of external and internal quality control assurance.
I am Abhipsa CHAKRABORTY and I am going to continue with the topic principle and application of microarrays, and I will mainly be focusing on two types of microarray. One is the protein microarray, another is the carbohydrate microarray. Carbohydrate microarrays. Carbohydrate microarrays have become powerful tool to study the binding events involving sugars, especially as a means to perform many low-cost, high-throughput analysis of carbohydrate interactions with a wide variety of biological targets, including proteins, nucleic acids, whole virus, and cells. Binding preferences, inhibition of interaction, enzyme activities, and structure-function relationships can be determined. Screening and characterization of antibodies as well as the identification of biomarkers can also be conducted. This slide usually shows the principle on which the function of the carbohydrate microarrays are based on. Here, the PAA biotin streptavidin is present on the ultrabind membrane and which undergoes the reductive amination, which lead to the following consequences like blocking lectins or the virus's binding, AP conjugated, anti-lectin or antivirus antibody, and the NBT or BCIP. Then quantification takes place in the ultrabind membrane and scanning also occurs, which produces a analytical graph, and they can be visualized by direct or by indirect fluorescent tagging. This slide shows the reagents and the buffers along with the apparatus that can be used for the high throughput carbohydrate microarray system. These includes the dilution bar buffer, which is a saline 0.9% of NaCl solution. There is a rinsing solution that contains 9-phosphate buffer saline with a pH of 7 along with 0.05% of T20. Lastly, there is a blocking solution which contains 1% of bovine serum albumin in the PBS, which is the phosphate buffer saline, along with 0.05% of T20, and also sodium nitrite. The apparatus usually includes the microspotting, which is the Cartesian technologies. Along with that, there are supporting substrate, which contains the fast slides. There are microarray scanning, which is the scan array 5000 standard BioJIT scanning system. Under the methods, the first method is the design and the construction of the chip. Here, an 8-chamber subarray system was being designed to construct the customized carbohydrate microarray. Each microglass slide contains 8 separated subarrays. The microarray capacity is approximately 600 microspots per subarray. A single slide is thus designed to enable 8 microarray assays. A similar design with array capacity of approximately 100 microspots is also commercially available. The next one is the microspotting of carbohydrate containing molecules onto the nitrocellulose coated glass slides. Using the Cartesian technologies, a high precision robot designed for cDNA microarrays, carbohydrate antigen of various complexities are picked up by deep quill pins from antigen antibody solution and printed onto the nitrocellulose coated fast slides in consistent amounts. The complementary AXYSX software of the Cartesian technologies is used to control the movement of pins during the dispensing and printing process. Then comes the immunostaining and scanning of microarrays. The staining procedure for the carbohydrate microarrays is basically identical to the routine procedure for immunohistology. Then lastly, there is the validation of microarray findings by conventional immunological assays. It is highly recommended that microarray findings will be verified using other experimental approaches. We usually confirm our results by at least one of the alternative immunoassays such as the enzyme-linked immunosorbent assay and the dot plot, the Western blot, the flow cytometry or the immunohistology. This slide shows the application for the carbohydrate microarrays, and here it mainly shows that the carbohydrate microarrays can be used for the vaccine design, for pathogen sensing, for enzyme specificity, and for inhibitor discoveries. Apart from these applications, the carbohydrate microarrays have been used recently for the rapid analysis of the glycan protein or glycan cell interaction and for the detection of pathogen. As a demonstration for its significance and versatility, the microarray technology has been applied in this effort to assay glycoside transferase activities. In addition, the carbohydrate microarrays based methods have been employed to quantitatively determine binding affinities between lectins and carbohydrates. Protein microarrays. Protein microarrays are used to determine the function of proteins as well as to monitor the interactions. The structure of the array allows the numerous proteins to be tracked in parallel. Protein microarrays were developed by utilizing the technology of DNA microarrays, which are commonly used to analyze gene expression. The requirements of protein microarrays are, however, more complex and necessitate material customization to make them suitable, even though they are not as commonly seen as the DNA microarrays. The protein microarrays are increasingly being used for multiple applications in the biosciences. This slide shows us the principle based on which the protein microarrays works. Here, the antibodies, the proteins and the peptides are fixed on a support slide. Then incubation occurs with the biological sample. In the third step, the antigen and the antibody reaction occurs in between the complementary molecules. The fourth step, the antigen-antibody reaction detection occurs. In the fifth step, the slide scanning and the image acquisition takes place, and at last, the analysis of the data occurs. The protein microarrays can be typically prepared by immobilizing the proteins into a microscope slide using a standard contact spotter or non-contact microarray. There are different methods for arraying the proteins. The most common type of protein array, the robots place large numbers of protein on their ligands onto a coated solid support in a predefined pattern. This is known as the robotic contact printing or robotic spotting. Another fabrication method is the inkjetting, a drop-on-demand, non-contact method of dispersing the protein polymers onto the solid surface in the desired pattern. The piezoelectric spotting is a similar method to the inkjet printing. The print head moves across the array and at each spot uses electric stimulation to deliver the protein molecules onto the surface via the tiny jets. This is also a non-contact process. Lastly, the photolithography is another method of arraying the proteins onto the surface. Light is used in association with the photomasks, opaque plates with holes or transparencies that allow the light to shine through in a defined pattern. A series of chemical treatments then enables deposition of the protein in the desired pattern upon the material underneath the photomask. There are 5 major areas where the protein arrays are being applied: diagnostics, proteomics, protein functional analysis, antibody characterization, and treatment development. Diagnostics involves the detection of antigens and antibodies in the blood sample, the profiling of sera to discover new disease biomarkers, the monitoring of the disease states and responses to therapy in the personalized medicine, the monitoring of environment and food. Digital bioassay is an example of using the protein microarray for diagnostic purpose. In this technology, an array of microwells on a glass polymer chip are seeded with magnetic beads, that is, coated with fluorescent antibodies, and they are subjected to targeted antigens and then characterized by a microscope through counting fluorescent wells. A cost-effective fabrication platform for such microwells arrays has been recently demonstrated, the bioassay model system that has been successfully characterized. The proteomics pertains to protein expression profiling, that is, which proteins are expressed in the lysate of a particular cell. Protein functional analysis is the identification of the protein-protein interaction, for example, identification of members of a protein complex. The protein phospholipid interaction, small molecule targets, enzymatic substrates, particularly the substrates of kinases and receptor ligands. The antibody characterization is characterizing cross-reactivity, especially the specificity and the mapping epitopes. Treatment development involves the development of antigen-specific therapies for autoimmunity, cancer, and allergies. The identification of small molecule targets that could potentially be used as a new drugs.
My name is SOUJIT. I would like to talk about SNP microarray. SNP means single nucleotide polymorphism. It refers to the single nucleotide at the same position in genomic DNA sequence. In general, a single nucleotide variation with frequency greater than 1% is called SNP. The SNP involves only a single base variation, which can be caused by single base transition or transversion. There is approximately 1 SNP per 1000 bases in human genome, and total number of SNP in human genome is around 3 million. SNP characteristics has make it more suitable for studies on genomic or genetic pleiotropy in complex trait and diseases, and population-based gene recognition than other polymorphic markers. That is why SNP has become the third generation of genetic markers. How does SNP work? SNP microarray uses nucleotide sequences as probes to hybridize with the tested DNA sequences, allowing qualitative and quantitative SNP analysis through single detection. Compared with traditional single-cell diagnostic method, SNP microarray is high throughput method which can perform thousands of reactions on the surface of oligonucleotide. There are general processes in a SNP microarray such as SNP chip fabrication, sample genomic DNA preparation, hybridization, fluorescence scanning, and signal scanning. So SNP chip fabrication. The prescribed oligonucleotide or cDNA chip are arranged orderly in a high density or glass carrier to make microarrays more cheap. The fabrication method of chips include light-guided in situ synthesis, chemical spray method, contact dot coating method, in situ DNA control synthesis, non-contact with micromechanical printed method, etc. Then simple genomic DNA preparation. Firstly, preparing the sample material, then the genomic DNA is extracted and is amplified by PCR. Finally, different fluorescent dyes are used to label it. Hybridization and scan, the last process. The genomic DNA labeled with fluorescence is hybridized with SNP microarray under suitable conditions. After being washed, a specific fluorescence scanner is used to scan the fluorescence. Then the computer images analysis is made to progress the data through target gene bio and bioinformatics analysis carried out. Applications. The SNP array is a useful tool for studying slight variations between whole genomes. The most clinical application of SNP arrays are for determining disease susceptibility and for measuring the efficacy of drug therapies designed specifically for individuals. In research, SNP arrays are most frequently used for genome-wide association studies. SNP can also be used for study genetic abnormalities in cancer. For example, SNP arrays can be used for the study of loss of heterozygosity or LOH. LOH occurs when one allele of a gene is mutated in deleterious way and the normal functioning allele is lost. A LOH occurs commonly in oncogenesis. For example, tumor suppressor gene help keep cancer from developing. If a person has one mutated and dysfunctional copy of tumor suppressor gene and in second functional copy gene gets damaged, they may become most likely to develop cancer. High density SNP arrays help scientists identify patterns for allelic imbalance. These studies have potential prognostic and diagnostic issues. Because LOH is so common in many human cancer, SNP arrays have great potential in cancer diagnostics. For example, recent SNP array studies have shown that solid tumors such as gastric cancer, liver cancer show LOH, and do non-solid malignancies such as hematogenic malignancies. These studies may provide insight into how these diseases develop as well as information on how to create therapies for them. Here are the list of applications such as gene discovery and mapping, association-based candidate polymorphism testing, diagnostic or risk profiling, response prediction, homogeneity testing and study design, gene function identification. These are the shortlisted applications of SNP microarray. That is why SNP microarray can be named as third marker for gene discovery. Thank you.
Hello everyone, my name is JAGATJIT SARDAR and I will be explaining about DNA microarrays. In this slide, I am going to explain about DNA microarrays. So what are DNA microarrays? DNA microarrays are solid supports usually of glass or silicon upon which DNA is attached in an organized grid fashion. Each spot of DNA, called a probe, represents a single gene. There are several synonyms of DNA microarrays such as DNA chips, gene chips, DNA arrays, gene arrays and biochips. Each DNA spot contains picomolecules of a specific DNA sequence known as probes. Each known gene or probe occupies a particular spot on the chip, and varying levels of fluorescent activity show different varying levels of gene activity when different types of genetic materials are introduced. Fluorescently labeled target sequences that bind to a probe sequence generate a signal. So in this slide I am going to explain briefly about how this works. So a DNA chip manufactured can contain thousands to hundreds of synthetic single-stranded DNA. Unknown DNA from a patient is separated into single strands, enzymatically cut and labeled with a fluorescent dye. The unknown DNA is inserted into the chip and allowed to hybridize with the DNA on the chip. The tagged DNA will bind only to the complementary DNA on the chip. The bound DNA will be detected by its fluorescent dye, analyzed by a computer. The red light is a gene expression in normal cells, a green is a mutated gene expressed in tumor cells, and yellow in both cells. So in this slide I am going to explain about the principle behind the DNA microarrays. So the core principle, that is the main principle behind DNA microarray, is hybridization. Samples are labeled using fluorescent dyes. At least 2 samples are hybridized to a chip. Complementary nucleic acid sequences get PR by hydrogen bonds, and then washing off non-specific bonding. Mainly, these are the main principle behind the DNA microarrays, and many more are there also. So this slide is about the design of a DNA microarray and the technique used for preparation of a DNA chip. So about the design of a DNA microarray system, so as you can see, first the sample was collected, then purification is done, then the recombinant technology is used, then coupling is done, hybridization is done, and then washed off is done, and finally the scanning, and then the normalization and analysis is done. And in the technique used for the preparation of a DNA chip, as you can see that there are many more processes, but the main concept of these processes are photolithography. So in this slide, I am going to explain about the different types of DNA chips or DNA microarrays. So there are mainly two types, that is cDNA based microarrays and oligonucleotide based microarrays. So what is cDNA based microarrays? It is a glass DNA microarrays which involves the microsporting of prefabricated cDNA fragments on the glass slide is used. And what is oligonucleotide based microarrays? And it is a high density oligonucleotide microarray, often referred to as chip, which involves in situ oligonucleotide synthesis. So in this slide I am going to be explaining about the applications of DNA microarrays. So there are many applications of DNA microarrays such as gene expression profiling, discovery of drugs, diagnostic and genetic engineering, alternative splicing detection, proteomics, functional genomics, DNA sequencing, toxicological research, etc. And I am explaining now about DNA sequencing. So first, what is done? From the liver or from the part from which you are going to extract the DNA was extracted from there. Then the library for the gene was prepared, where hundreds and millions of genomic DNA fragments are there and collected on a surface. Then single genomic fragment was opted out from there, and then make thousands copies of that same single genomic fragment, and then genomic samples are then sequenced in a machine, and then the computerized data was analyzed and the data was taken out. So in this slide I am going to be explaining about the advantages and disadvantages of DNA microarrays. So let's take the advantages first. So first point is provide data for thousands of genes. Second is one experiment is done instead of many, so it takes less time. Fast and easy to obtain results. Huge step closer to discovering cures for disease and cancer. Different parts of DNA can be used to study gene expression. So I would like to take this point of huge step closer to discovering cures for disease and cancer, because if someone is being diagnosed with a disease or some cancer, and that person is given medicines, but the medicine is not acting properly because that person is having some specific kind of disease which is creating problems in him or her DNA, from him or her DNA. But if the medicine is not specific for that person's DNA, then that will take a much longer time to be cured. So by this point or by this process, we can give that person a specific kind of medicine which will be more useful for curing cancer or the disease for that person. Now consider the disadvantages. So first is the biggest disadvantage of DNA microarray is that they are very expensive to create. Second is the production of too many results at a time requires a long time for analysis, which is quite complex in nature. And last of all, the DNA chips do not have very long shelf life, which provides to be obtained another major disadvantage of the technology. So last of all, the conclusion. So DNA microarrays are one of the most effective invention ever developed. A DNA microarray is a test that allows for the composition of thousands of genes at once. Microarray technology uses chip with attached DNA sequences as probes for gene expression. Any DNA in the sample that is complementary to a proper sequence will become bound to escape. Microarray technology is the most useful and powerful when it is used on species with the sequence to genome. The microarray chip can hold sequences for every gene in the entire genome, and the expression of average in it can be studied simultaneously. Gene expression data can provide information on the function of previously uncharacterized genes. So as we can see, it is a very helpful technology, but it has some advantages and some good advantages and better advantages, but it had some disadvantages too. So that's all from my side. Thank you.
This is Swatiparna. Let's talk about antibody microarray, a complete microarray based system for profiling protein expression in biological samples, used to compare two biological samples to measure the relative differences in protein expression. The microarray consists of hundreds of monoclonal antibodies covalently bound in an ordered layout to a glass slide, a protein which can be synthesized in pure form by a single clone population of cells. These antibodies can be made in large quantities and have a specific affinity for certain target molecules called antigens, which can be found on the surface of cells and those that are malignant. The array can be used as a means to correlate specific proteins with physiological or pathological process of interest by comparing hundreds of proteins at a time. It is used for toxicity testing, disease investigation, and drug discovery. Let's know about the antibody array procedure. Extraction of total cellular protein from biological samples of interest. Labeling of extracted protein with fluorescent dyes such as CY5 and CY3. Removal of unbound dye. Incubation of labeled protein with the array. Scanning of the array and the analysis of the results. Let's talk about phenotypic microarray. Phenotypic microarray technology is growth of microorganisms, either pure culture, community, or consortia, in multi-well plate, each with a different test component in each well, enabling a screen of the phenotypic characteristics of the test culture. This technology allows for culturing in small volumes, providing opportunity to perform many parallel assays in a compact size. Examples of well components could include, but are not limited to, carbon, nitrogen, phosphorus, sulfur sources, amino acids, pH or NaCl tolerance, or antibiotic resistance. Some highlights of phenotypic array. Phenotypic screening rapidly assesses a wide range of growth conditions. Growth assays can be adapted for anaerobic and extremophile growth conditions. Phenotypic arrays assess substrate utilization in environmental communities. Cluster heat maps are effective in computing and visualizing PM data. Improved results are obtained through parametric curves rather than summary values.
Now let us see something called as interferometric reflectance imaging sensor, or IRIS. Interferometric reflectance imaging sensor, or IRIS, formerly known as spectral reflectance imaging biosensor, SRIB, is a system that can be used as a biosensing platform capable of high output multiplexing of protein-protein, protein-DNA, and DNA-DNA interaction without the use of any fluorescent levels. The sensing surface is prepared by robotic spotting of biological probes that are immobilized on functionalized silicon or silicon dioxide substrate. IRIS is capable of quantifying biomolecular mass accumulated on the surface. To perform a measurement, the sample is illuminated with the multiple different wavelengths from either a tunable laser or different colored LEDs, typically speaking a relatively narrow bandwidth optical source. The reflection intensity is imaged using a CCD or CMOS camera. By using interferometric techniques, nanometer changes can be detected. Applications of IRIS include microarray format immunoassays, single nucleotide polymorphism or SNPs detection, pathogen detection, biodefense monitoring, kinetic analysis of biomolecular interactions, and general biomolecular interaction studies for research applications. So, the technique, though limited at present in its applications due to the high cost factors, may widen its prospect once there's increase in availability of commercial products. The ability to record large number of old samples and analyzing them for various genetic alterations help in understanding the concept of molecular biology. Microarrays hold much promise for the analysis of disease in oral cavity. Classification of oral diseases by DNA, RNA, or protein profile will greatly enhance our ability to diagnose, prevent, monitor, and treat our patients. Currently, microarrays are primarily a research tool. Microarrays promise a more biologically based, individualized, and vastly improved standard for oral care, which will have great clinical impact on the way dentistry will be practiced in the near future.
So this is the end of our presentation on the topic Principles of Microarrays and its application. Thank you.